Okay, so today we're going to be talking about how living things capture free energy. Make sure that you understand the idea of free energy. When I say things like delta G, which I'm going to say a lot today, you should know what that means. The change in free energy. And you should also know what it means for delta G to be positive or delta G to be negative. All right, so let's jump right in. We got a lot to talk about today. So first of all, we want to talk about that all organisms can be classified into two groups. They are either autotrophs or heterotrophs. Autotrophs are things that are able to capture free energy from basically for themselves. They don't have to rely on any other living things in order to survive. Best example of this would be plants. So you think of the fact that a plant is able to do photosynthesis, and so it captures free energy. So if we think about what that means for a second, you've got sunlight coming down, hitting the plant, and the plant is going to take the energy from the sun, and it's going to store it. Whenever we store energy, we're going to have a positive delta G. So for autotrophs, they're able to do this thing where delta G is greater than 0, and they're actually storing energy. Now they're also able to then release that energy. They store the energy as sugar, and then they're able to use that sugar later to energize themselves. And so in that case, when they're releasing that stored energy, delta G would be less than 0. But autotrophs are able to do this. They're able to take energy from the environment and store it as energy that they can use. There is another kind of autotroph as well, and that would be an organism that is what we call chemosynthetic. So organisms that are chemosynthetic can actually use small molecules just present in the environment in order to do what we call chemosynthesis. Which is another situation where you're actually able to take the energy from those chemicals and store it. So as far as the organism is concerned, it is storing energy, so delta G is going to be greater than 0 in that case also. Now heterotrophs are not able to take energy from the environment. What they have to do, if you think about an animal like maybe a cow, this is my best attempt at drawing a cow right here. That's as good as it gets. Bam. Well, this cow has to go ahead. He cannot make his own energy from chemicals lying around in the environment. He cannot absorb energy from the sun. So what he has to do is he has to find a plant which has already stored energy, and he has to steal the energy from that plant by eating it. So the energy transfer is that he's stealing the stored energy from the plant and using it for himself. In this case, the cow is actually releasing the stored energy of the plant, and so for the cow, for heterotrophs, they are only able to do situations, basically reactions, overall where delta G is greater than 0. So they're only able to access and use stored energy from other living things. If the plants all died, then all the heterotrophs would die because they would have nothing to eat. So we're going to be focusing today on autotrophs, and we're going to be focusing specifically on the process of photosynthesis and how that works. So let's start with an overview of photosynthesis. First of all, this picture shows you the most important thing in photosynthesis, which is a chloroplast. So chloroplasts are organelles. They're little organs inside of a cell that allow plant cells to do photosynthesis. These are unique to plant cells. They're not found anywhere else. And they do a very complicated chemical reaction, which is, first of all, they do what's called the light reactions of photosynthesis. These are called the light reactions because, as you notice from this diagram, they're powered by light. And then after they've done the light reactions, they use the products from that in order to do another cycle called the Calvin cycle, which is called the light-independent reactions because they don't require light. We're going to focus on the light reactions first, and then we'll go to the Calvin cycle. So, like we said, light-dependent reactions happen inside of this thing right here, which is a chloroplast. More specifically, they happen inside of little spheres or disks that are stacked inside of the chloroplast called thylakoids. So each of these little green spheres, or little green disks, I should say, is a thylakoid. And they're the ones responsible for doing the light-dependent reactions. So if we were to look at the membrane of the thylakoid, the little outer surface, that's where all the action happens. And so this diagram shows the membrane of the thylakoid right here. You can see that's labeled. This is the membrane, the phospholipid bilayer. And inside of that membrane are a bunch of proteins. We call these proteins photosystems. And their job is to actually absorb light and use it. So we're going to zoom in on this diagram real quick and learn a little bit more about those photosystems. So we're going to start with photosystem 2. That's a little bit contradictory. We're going to start with photosystem 2 and go to photosystem 1. Photosystem 2 is number 2 because it was discovered second, not because it's the second most important or it comes after photosystem 1 in the process. We actually start with photosystem 2. So what happens in photosystem 2 is that you have these guys. These are pigments with a very well-known name. They're called chlorophyll. All right, and those chlorophylls absorb photons. Photons are particles of light. When they absorb that photon, an electron becomes energized. All right, so we get and the source of that electron is a water molecule, H2O. So you go ahead and actually split. The first thing that happens, or one of the first things that happens, is water molecules get split into oxygen and some extra hydrogens. Now, one of the electrons is split off by photosystem 2, and that electron becomes very energized, and it's that energized electron that powers the next stage. So photosystem 2's job is to take light and use it to energize electrons from water. All right, so the next step then is that electron gets passed along through a bunch of different molecules. So the electron goes over here to this molecule, and then over here to, these are membrane proteins, to this protein, and over here to this protein, and then over here to another protein. Each time it gets passed along, some energy from the electron is lost. So it's releasing a little bit of energy. So we would say that that energy is going to be used for something, and we'll talk about what that energy is used for in just a second. But this process of the electron being passed from one protein to the next, losing energy, is called the electron transport chain. All right. So after you go through the electron transport chain, you're going to get, that electron is going to get passed to another photosystem. So it gets passed from the electron transport chain over to photosystem 1, and the same thing happens all over again. So that electron gets energized once more. You can see here the electron is, and it gets energized by another photon of light, and then that high energy electron gets passed to other molecules once again. So you actually go through the electron transport chain twice, each time releasing a little bit more energy from that electron. Eventually, the electron has run out of energy, and it just needs to be picked up so that it can be transported back to photosystem 2. And the thing that picks it up is called NADP+. And when it picks up that electron, it becomes NADPH. This is what we call an electron receptor. Or electron acceptor, one or the other. So NADPH just picks up that electron and then carries it back to photosystem 2 so that it can be reused. All right, let's look a little bit more closely at what happens in the electron transport chain and why that's occurring. So we're going to be looking at this diagram. This is actually a graph, and it shows basically energy on the y-axis. So you start off with the electron in a water molecule, and that electron gets broken off by photosystem 2 and placed into a chlorophyll. Light comes along and hits that chlorophyll, which energizes the electron. So you can see the electron moves up in energy to here. Now the electron transport chain, the molecules or the proteins are gradually releasing the energy of the electron. So that's a gradual release. Now, a gradual release of energy is important because it's more efficient than simply releasing all the energy at once. You have a better chance of the chloroplast being able to use that energy if it's a gradual, steady release of energy. And then that electron gets passed to another chlorophyll after traveling through the electron transport chain. It gets hit with another photon, re-energized, and then passed through another electron transport chain, and ultimately gets picked up by NADPH. And like we said, NADPH is going to take that electron and transport it back over here so that it can be reused. So we were just saying that one of the things that happens during the electron transport chain is that energy is being released. Well, this diagram does a better job of showing us what that energy actually is used for. So we have the photosystem 2 is right over there on the left, and it's kind of cut off. You're not able to see the whole thing. But there it is, photosystem 2, and then photosystem 1 is right here. So the electron transport chain that we're going to be talking about is in between those. So we said that the energy of the electron is being released by the electron transport chain, and what it's doing is the electron transport chain is taking protons, these little H pluses, and it is driving them from outside the thylakoid into the thylakoid. So each of these molecules is moving protons across the membrane. Now you'll notice that means that you're ending up with a situation where there's lots of protons inside the membrane and very few protons outside the membrane. That means that the protons have a strong desire, there's a big energy payoff, if these protons would move back outside. They do not want to be cramped inside the thylakoid, they want to spread out. And part of the reason for that is because they're all positively charged, and those like charges repel each other. So you've created a situation where there's lots of protons that want to move outside. And when they move outside, that's actually going to release some stored energy. So when you move those protons, delta G is going to be less than 0. Energy is actually going to be released by moving those protons. So the chloroplast takes advantage of that. It uses a molecule, which is right here in purple, called ATP synthase, or synthetase. And what ATP synthetase does is it allows these protons to move through. But when the protons move across the membrane, it takes the free energy that's being released and it uses it to generate ATP. Right there. And it generates a pretty healthy amount of ATP. So in the next video, we're going to be talking about what that ATP does and what happens in the rest of photosynthesis.